In this presentation, we are going to see about bacteriophage. It is a virus living inside the bacterial cells. It is also called as bacteria eating agent. It was first discovered by the scientist called Twart in the year 1915. The common bacteriophage is T4 bacteriophage. Coming to the structure of the bacteriophage, this T4 bacteriophage is a tadpole shaped. It consists of three parts: a head, a neck and a tail. The head is a polyhedral in shape and covered by a protein coat called capsid. This capsid is made up of 2000 protein subunits called capsomeres. A double-stranded DNA is present inside the head and the neck is very short. It contains collar. This collar connects the head and the tail part of the bacteriophage. The tail part consists of a central hollow core tube which is covered by the protein sheath. The free end of the core tube has a hexagonal end plate. This end plate has 6 spikes and 6 fibers. These 6 spikes and 6 fibers are responsible for the attachment of the bacteriophage on the surface of the bacterial cell wall. Let's move on to the multiplication of the bacteriophage. Bacteriophages are capable of multiplication only within the bacterial cell. They remain inactive outside the bacterium. For example, T4 bacteriophage infects and multiplies only inside the bacteria called E. coli. The multiplication of the phages takes place in 5 steps. The first step is attachment of the phage on the bacterium. Second step is injection of the phage DNA into the bacterial cell. The third step is disruption of the bacterial metabolism. The fourth step is assembly of phage particles inside the bacterial cells. And the last stage is lysis of the bacterial cell. The first stage is attachment of the bacteriophage on the bacterial cell wall. When the phage and the bacterium come close together by random collision, the phage attaches itself to the surface of the bacterium. The tail fibers present in the phage, that is in the end plate, select the specific site on the bacterial cell wall and attaches with it. The next stage is injection of DNA. After attachment, the tail secrete an enzyme called lysozyme which dissolves the bacterial cell wall to make a hole. The contractile sheath contracts and this forces the tail core into the bacterial cell wall through the hole. The phage DNA is then injected into the bacterium through the core. The next stage is disruption of bacterial metabolism. In this stage, the phage DNA disturbs the bacterial metabolism and synthesizes its own DNAs and proteins. The synthesis of normal bacterial protein stops and the phage DNA replicates by using free nucleotides and produces many copies of phage DNA. The next stage is assembling of the bacteriophage. During this stage, the phage proteins form subunits, tail sheaths, tail fibers and end plate. The subunits form into head. Each DNA copy enters into the head and becomes tightly packed. The tail sheath and the end plate join together to form a tail. This tail attaches to the head. Thus, about 150 to 300 phages are produced in a bacterium. The last stage in the multiplication of the bacteriophage is lysis. As many phages have been produced inside the bacterial cell wall ruptures and the phage particles are released free. Each phage particle, which in turn infects other bacterium, and the cycle continues. Thank you.